So this section of notes is on cells. So if you look at this picture, we have several different types of cells. So you have a plant cell over here. That's probably the one that caught your eye. This is a nerve cell. These are red blood cells. And this is a zygote in the process of splitting and forming into new cells. So this shows like an embryo when it was the sperm and egg came together, and then it splits and splits again, and eventually you have exponential growth. We'll talk more about that when we get to that unit though. But here's just a few pictures of some different cells, since we're discussing cells. So just some general information. Cells, if they're given nutrients and given the right environment in a lab, they can last for years outside of the body. So this is really cool. There's actually a book that talks about Henrietta Lacks. I think that's how you spell it. And HeLa cells. I'm trying to remember the exact name. It's like The Immortal Life of Henrietta Lacks. So if you're into reading, and y'all might read that at some point in some English class, but it's called The Immortal Life of Henrietta Lacks, and it talks about this woman named Henrietta Lacks, and she's one of the first people who they were able to successfully get cells to live outside of the human body for any length of time. And they were actually her cancer cells. So long after she died, her cells were still being used. Anyway, so cytology, cyto is cell, logy is study of, so cytology is the study of cells. So 3 parts of the cell theory. This is just a review from our first unit. Remember, all living things are made of one or more cells. The cell is the basic unit of structure or shape, and function, that would be its job in organisms. And of course, all cells come from already existing, pre-existing cells. So there are 2 main types of cells, prokaryotic and eukaryotic cells. So the first one we're going to discuss is prokaryotic. So a prokaryote is a prokaryotic cell. So pro means before, and karyo means nucleus. So prokaryotic cells supposedly existed before cells had nucleuses or nuclei. So these are the simplest type of cells. They are very small, much smaller than a eukaryotic cell. One of the reasons why they're so small is because they're very simple. There's not a lot inside of them. They don't have all those organelles like the mitochondria and chloroplast and the Golgi body and all those things. So they don't have membrane-bound organelles. So again, they do not have a nucleus. So if you look at evolution, some of the accepted theories of evolution, they believe that prokaryotic cells would have been the first to evolve. So anyway, because they have no nucleus, it's very important you know where DNA is found. And so because there's no nucleus, the DNA is just floating in the cytoplasm. So prokaryotic cells usually contain a cell wall, a cell membrane, cytoplasm, and ribosomes. And of course, like I said, the DNA will be found floating in the cytoplasm. So there's not a lot to them. Basically, on the inside of a prokaryotic cell, you have your cytoplasm, some ribosomes, and some DNA. And then they have every cell has a cell membrane. And usually they have a cell wall on the outside of that cell membrane, and some might even have what's called a capsule. Anyway, our only example of prokaryotic cells are bacteria. If it's not bacteria, it's not prokaryotic. Bacteria are the only example of a prokaryotic cell, okay? So make sure that you know that. Everything else that is a cell is eukaryotic. So again, I mentioned that outside of the cell wall, sometimes you'll have what's called a capsule. So it will be on the very outermost layer, and not every bacteria cell, not every prokaryotic cell will have a capsule, but if it does, it will provide extra protection because bacteria are typically unicellular. Now, the plasmid is what we call bacterial DNA or DNA in a prokaryotic cell, and it's circular DNA. So it's still a double helix, but that double helix makes a circle. So this is where the genes for the bacteria or the prokaryotic cell is found. So anyway, it's involved in growth and metabolism, the plasmid is. Then you have metabolism. So metabolism, again, all living things are going to utilize energy of some sort. So prokaryotic cells do have a metabolism. So these prokaryotic cells are going to make, store, and transport chemicals. They're also going to utilize energy. Okay, so some prokaryotic cells can make their own food because there's some bacteria that are photosynthetic. Some may be even chemosynthetic, and we'll talk about what that means in a different unit, but it's basically chemicals. They use chemicals to make food, and then some they may eat, so they may be what we call heterotrophs, just like you and I. And they could also be a decomposer, which a decomposer is a type of heterotroph. So there's three main shapes you might find prokaryotic cells have. So again, prokaryotic cells, our only example is bacteria, so that's why it says bacterial shapes. So you have spirillum, which is a spiral shaped. Spirilla is plural. Bacillus, that is rod shaped. So bacilli is plural. And then you have coccus, which is a round or spherical oval type shape. And cocci is going to be the plural for that. So if you see any of these terms, it is referring to prokaryotic cells or bacteria. So this is a picture of a prokaryotic cell. So in your notes, if you wanted to draw this, you could, but I want to point out a few things. So and every cell is going to look a little bit differently, but typically this will be a normal prokaryotic cell. So you're going to have your cytoplasm, of course, filling the space on the inside of the cell. So in this case, they've called the DNA the nucleoid. That is not the same as a nucleus. Notice there's no little ball. The little ball is the nucleus, okay? And then you have ribosomes. That's pretty much it on the inside of this cell, which is why it's smaller and more simple. So then you have your cell membrane, okay? That's this gray layer here. Then you have your cell wall. Then you have your capsule. So, again, you don't always have those things, but bacteria usually do because they're unicellular and they, need something to protect them. So, usually, again, they're unicellular, so they need some way to move. So, a lot of them have flagella, which is this tail-like structure. Some of them might have cilia or other things to help them move, but usually flagella and cilia are the two common ways that bacteria would move. And then sometimes they have something called pili. So we're going to talk a little bit more about pili. But these pili basically help the bacteria stick to surfaces. And this would be why we need to use friction in addition to cleaning products like soap or disinfectants to clean our hands. You can't just put soap on your hands. You have to actually scrub your hands and use that friction to help get the bacteria off. Same thing with your teeth and brushing your teeth and flossing. You have to use the toothbrush to scrub in addition to the toothpaste or the mouthwash. Okay, so think about like alcohol-based mouthwash. Typically alcohol is going to kill bacteria, but you do need that motion, that friction. So that's why they would have you swish the mouthwash to help give a little friction to it. And this is why you can't just use mouthwash. You got to actually brush your teeth. The toothbrush doesn't get everywhere, so therefore you have to floss. And flossing gets in the spaces that the toothbrush cannot to help physically move the bacteria off. Same thing when you're cleaning your desk at school. We have to apply some friction to it. I can't just spray it. We have to wipe it because we have to get those pili off that are sticking to the surface. So in prokaryotic cells, the DNA is found A, floating in the cytoplasm, B, attached to the ribosome, C, surrounded by the nuclear membrane, or D, contained in the vacuole. So very important you pay attention to what kind of cell it is, because later on we'll start mixing in eukaryotic cells. So in a prokaryotic cell, though, the DNA will be found, hopefully you said, floating in the cytoplasm. Which of the following are prokaryotes? Okay, remember I told you there's only one example of prokaryotic cells. Is it protist, plants, animals, fungi, or bacteria? Just making up stuff over here. So the only example of prokaryotic cells should be bacteria. So all these other things, and animals too, which is not even on here that I said, these things will be eukaryotic. And only bacteria are prokaryotic. So here we are to eukaryotes or eukaryotic cells, same thing. So eukaryotes, I'm going to break the word down again for you. EU or U means true. Karyo, again, hopefully you remember, means nucleus. So eukaryotic cells have a true nucleus. So they actually have a nucleus. And that's going to be the major difference is going to be having a nucleus. But they also have organelles, those membrane-bound organelles. So two major things you need to understand about eukaryotic cells is they have a nucleus and they have membrane-bound organelles. Now, because they have all this extra stuff, they're more complex. And they're also larger. Okay, if you recall, I had 1 to 10 microns or micrometers for a prokaryotic cell, but eukaryotic cells are 10 to 100 microns or micrometers, so they're much larger. Anyway, examples are going to be everything else that's considered alive: plants, animals, fungi, protists. Remember, viruses are not alive, so they are not cells. So, again, your eukaryotic cells have a true nucleus, and that is where the DNA can be found, is in the nucleus. There are other internal compartments, so the insides of the cell are called organelles. And each organelle has a different function, and those organelles allow the eukaryotic cells to have a different function than prokaryotic bacteria cells. So, here's a question for you. What type of cell is pictured below? Prokaryote, eukaryote, plant, or animal? Well, hopefully you noticed the nucleus, and in noticing the nucleus, you would have realized that this was a eukaryotic cell. Now, typically a plant cell is not going to have a flagella, so this is probably an animal cell, but it could also be a protist cell with a flagella. And the picture's not good enough for us to know that, nor would I ask for you to know that. Which of these types of cell has a nuclear membrane? Well, if it has a nuclear membrane, that means it has a nucleus. So what type of cell has a nucleus? A spirillum, coccus, liver, or bacillus? Well, hopefully you chose liver, because livers is something you have, animals have, and animals are eukaryotic. That was an example we gave. Okay. In addition to that, we said bacterial shapes were spirillum, coccus, and bacillus. Another way you could have answered this is by picking the outliers. Spirillum, coccus, and bacillus, those are weird words, so those are outliers compared to the liver. You can tell the liver does not belong with this grouping of words. So that's just a test-taking strategy. Coded information for heredity and protein production is contained in DNA, found in the eukaryotic cell structure called the nucleus, plasma membrane, endoplasmic reticulum, or Golgi apparatus. So hopefully you know that DNA in the eukaryotic cell is in the nucleus. So notice I can ask you eukaryotic or prokaryotic, so you need to be very careful with reading the question. So here's a picture of a few different structures and also an animal cell. Okay, so if you look here, you have cilia. Okay, you can't see that very good. There should be a cilia. So all these the little hair-like structures, that's actually the inside of like your trachea, your windpipe that goes to your lungs. So if you smoke, then you probably don't have these cilia. So what those cilia do in your lungs is they brush out like any dust or dirt or pollen that we might be breathing in, or pollution that you breathe in. So they help sweep those out of your lungs so you don't have to cough so much. So smokers have to cough so much because the cilia is singed off. The good thing is if you stop smoking, then it grows back. So this is one of the many reasons why smoking is not good for you. And that's any kind of inhaling things that you shouldn't inhale. So that would include vaping too. So don't do that. These are flagella. Flagella are the little tail-like structures that you see. And a lot of people they think that cells only have one flagella, but that's not always the case. Yes, human sperm typically only have one flagella, but human sperm are not the only cells with a flagella or flagellum. Many protists they're unicellular organisms that live in the water. They're microscopic. A lot of protists, they have multiple flagella. And then bacteria, of course, can have multiple. They can also be on opposing sides. Okay, so they might be on this side and this side. So they could spin in a circle really easy, I guess. Anyway, so this is just a picture of an animal cell. So anytime I show you these pictures like this one in the prokaryotic cell, you need to be prepared to be able to label it. Now, one thing that's not showing up here, but it is in the PowerPoint, but I'm going to go ahead and label it, is the mitochondria. Okay, so that's these structures. That's the mitochondria. And I know it's the mitochondria because it looks like a maze on the inside. And so we'll be talking more about those later. But anyway, you have your nucleus in the center. All the little dots on the outside of the nucleus are the nuclear pores. That way the RNA can leave the nucleus. Touching the nucleus, you'll have your endoplasmic reticulum. And so the endoplasmic reticulum will have a rough section with ribosomes on top of it. That's the dots. And then a smooth section with no ribosomes. And usually the endoplasmic reticulum goes from the nucleus out towards the cell membrane. And then you'll have the Golgi bodies, Golgi apparatus, Golgi complex. It's all the same thing. It's the Golgi. It'll be close by too. And you'll understand why later on when we talk about those structures. Then because this is an animal cell, it has centrioles. They're usually in pairs close to the nucleus because they help when the cell gets ready to divide. Lysosomes are also something you see only in animal cells, and they help with digestion. So just a few structures to look at there. What structure could a cellular organism use to move through its environment? So would you use a flagellum, cell wall, mitochondrion, or lysosome? Hopefully you picked flagellum, the tail. So cell size and shape. That funny looking U stands for micro. So micrometers are typically the unit that we use to measure cells. They are 1/1,000,000th of 1 meter or 1,000th of a millimeter. A millimeter is going to be the smallest marking on a ruler. So because cells are so teeny tiny and they're microscopic, we use micrometers to measure the size of cells and organelles. So cell size and organelle size is measured in micrometers. And again, they're microscopic. So organelles. So cell fractionation is a way to study organelles. So you're probably wondering, like, how do they know all these organelles do all these things? And they've used cell fractionation by separating your major organelles of cells in order to study what each organelle does. Now, the basic definition of an organelle is a structure carrying out a specific activity or specific function. So, for example, lysosomes, which are only in animal cells, they digest things. They break down things. So they're kind of like the stomach of the cell. So movement. I did place that one question in kind of not the best spot, but that's okay, y'all. Y'all still, I think, understood it because we talked about flagellum twice on 2 different pictures. So anyway, these are ways that cells can move. So one way that cells can move or move through things across the surface or into the cell is through microvilli. So microvilli are considered finger-like. They're short finger-like projections. And they're on the surface of the cell membrane. And what they do is they increase the cell's absorptive area. Now, here's a picture of it. It's not the best picture, but what it is like, for example, your small intestine absorbs most of your nutrients and things like that when you eat food. So if this is the surface, and if I have a flat surface here, then it can't absorb that much. But if I have a surface surface this does like this, now I have lots of extra space, lots of extra spots to absorb all those nutrients that you took in when you ate in those carbohydrates and proteins and lipids, and you took in your vitamins, etc. So that's going to be a lot more spaces. So it increases the cell's absorptive area. Cilia are those short hair-like projections. We usually see hair-like as a way to describe cilia, and they're on the surface of the cells, and what they do is they transport things across the surface of the cell. So you can think of them as like a broom. So like I said, in the trachea, like through here, your windpipe, it's going to line all that. You also have it in a few other places, but anyway, so it's going to sweep out stuff that got into your lungs that you don't want there. Then you have flagella. So that is, sorry, it's covering it up. Flagella or flagellum. Flagella is plural. So it's a whip-like or tail-like structure. Okay, and I think most people know what a flagella looks like. So this picture is several different flagella. And then you have pseudopods. Now, pseudopods are really cool. So pseudopods are typically in an amoeba. Amoeba is basically known as a shapeless blob, pretty much. They are a shapeless animal-like protist. So they're a protist. They're unicellular usually, and a lot of the times they live in water. So if you look, these cells on this side, they all have kind of a distinctive shape. You notice the shape with them, but the amoeba, they don't have a distinctive shape. They're kind of all over the place with their shape, and every one is going to be shaped a little bit differently. So if you notice, the shape is there's like not a distinctive shape. So all these little outpouchings are going to be called pseudopods. So if you recall, pseudo means false and pod means foot. So this is a false foot, and that is used for movement for the amoeba. So this is considered an extension of the cytoplasm. So then you have pili. I mentioned pili earlier when we were looking at the picture. Pili helps those bacteria cells stick to surfaces. So if I'm looking at them in a drawing, they do look very similar to cilia, but the function is to stick to surfaces. So another example of, I already mentioned like bacteria on your teeth, which is why we brush teeth to avoid cavities, because cavities are caused by bacteria. I also mentioned bacteria like on a desk, or like while we wash our hands, we have to like physically scrub them. Also Germ-X, if you use Germ-X, you have to physically like rub it in because of that friction. So anyway, with pili, another example is like strep throat. So I don't know if this has ever occurred to you, but when you have strep throat, that bacteria is sitting in the back of your throat. Well, you're swallowing all day. You're swallowing your spit, you're breathing, you are eating, drinking all day long. And so when you have strep throat, why aren't the bacteria being washed into your stomach and the acidic conditions in your stomach getting rid of them and killing them? Well, that's because the bacteria has pili. So the pili is causing them to stick in that spot in your throat, and then if you get enough of them there and your body can't fight it off, well, then now you have strep throat. So now we're getting into the parts of the cell. So if you'll notice the title, both plant and animal cells contain these organelles, and of course that's also protists and fungi as well. A lot of people just mainly think about plants and animals. So they both have these organelles. So the cell membrane, every cell has a cell membrane. The cell membrane also may be called the plasma membrane. So the cell membrane is going to protect the environment inside of the cell compared to outside of the cell. So the inside of the cell environment versus the outside environment. Okay, so it's surrounding the cell and it protects the cell because it's only going to allow certain things in and out. And it keeps that cell separate from the environment. So let's say you put a cell in super salty water. Well, you don't want the cell to lose water. So the cell membrane is going to protect it and keep those two environments separate. Hopefully, if it's doing its job. Now, like I mentioned, it kind of chooses what goes in and out. So it's considered selectively permeable. So it selects what enters and leaves the cell. So it keeps out some molecules and it allows others to pass. Now, small things like water, carbon dioxide, and oxygen gas, soluble or easily dissolvable end products of digestion are going to go through easily. Bigger molecules like glucose, ions are not going to pass through as easily. So the cell membrane is going to regulate and control what goes in and out of the cell, like gases. For example, oxygen. If your body has too much oxygen, well, the cell membrane is going to get rid of some oxygen. If it has too little, then it's going to take it in, and you'll start breathing more. Nutrients, same thing goes for, carbohydrates and different minerals and vitamins. Waste also. So this control keeps the cell alive and well. It's basically going to help maintain homeostasis because, like, if the cell needs water, then the cell will take in water. If it needs to get rid of water, then the cell will get rid of water. So the cell membrane is a major component in controlling homeostasis and keeping the cell alive and well. If it was not selectively permeable, everything on the outside would just come inside. This is a repeat. Sorry about that. So anyway, the cell membrane is made of the two P's. The two P's, phospholipids and proteins. So the phospholipid, we did learn about it in the previous unit. It's very important for this unit too. So make sure you understand the cell membrane and what it's made of. So phospholipid is a lipid, and it has a phosphate group on the head, and then it has two fatty acid tails. Okay, so the fatty acid part is the lipid part. The head is polar, so the phosphate part is polar. And of course, we know that fatty acids are nonpolar. And hopefully this is a refresher from the last unit. So then you have your other P, proteins. So again, the cell membrane is made of phospholipids and proteins. That's the main things. There are a few other things that can be in the cell membrane, but those are the main things. So you have lots of different types of proteins located kind of interspersed into that phospholipid bilayer. So transport proteins are going to help transport things in and out of the cell. So they're going to help move things in and out of the cell. You also have recognition proteins on the outside of the cell membrane, and they are receptors to recognize if the cell belongs. So for example, a bacteria cell is not normal to be in your body. So these recognition proteins can notice a bacteria cell and notice that it's not supposed to be there. Or it can notice one of your own personal red blood cells and know that it is supposed to be there. Anyway, then you have integral proteins. Integral proteins penetrate the membrane. Okay, so they go all the way through the membrane. They're not just sitting on the outside. They go all the way through both layers of the lipid bilayer. And so basically they're considered like pipelines or conduits to transport things in and out of the cell. So a lot of these proteins are going to help move things in and out of the cell. Then you have peripheral. Peripheral is like on the outside. So if you think of your peripheral vision, that's your vision around the outside, not your center vision. So peripheral proteins are going to be attached to either the inside or the outside surface, and they do not go all the way through. A lot of the times these proteins are enzymes, so they might serve an enzyme function or a structural function. So which of the following is usually measured in micrometers? Okay, well, meters is going to be some sort of length or height. Mass would be grams, so it's not mass. Sound would be like decibels. It should be dB, sorry. So it's going to be either length or height. So if it's microscopic, that's not going to be a tree. It's going to be B, organelle length. The unique properties of a cell membrane arise from the arrangement of two layers of organic molecules called what? So the cell membrane, what are the two layers made of? Cellulose, nitrates, nucleic acids, or phospholipids? Hopefully you remember the phospholipid bilayers. So here's a picture of the cell membrane. So if you notice, you have your one phospholipid highlighted here, and you had your hydrophilic or polar head here, and it loves water. And then in the center you have, here, let's change color. So in the center you have the tails, which are going to be nonpolar because they're lipids, they're fatty acids, and so they fear water. Okay, so notice you have the two layers here. So this would be a membrane protein, and notice this goes all the way through, so that's probably an integral protein. So anyway, this is just a picture of the cell membrane showing you that phospholipid bilayer. So the whole outside and the inside. So if this is the inside of the cell likes water, and then this is the outside of the cell. The outside of the cell likes water, but if it tries to go through the membrane, it typically does not like polar or substances like water. So they are usually going to travel through the protein rather than squeeze in through here. This is also a small space. That's a small space, so larger molecules are also going to go through that protein part. Look at my beautiful art. The part of a eukaryotic cell that allows it to remain separate from the outside environment is what? The cell membrane, ribosome, cytoplasm, or Golgi vesicles. So, remains separate from the outside environment, that should be the cell membrane. Okay, so this slide we're talking about cytoplasm. So, cytoplasm is a jelly-like or fluid-like material, and so what it's doing is it's filling up all the extra space inside of the cell. So you have the nucleus inside of the cell membrane, and then all your organelles, and if it's not an organelle or the nucleus or some other cellular structure, then the cytoplasm is going to fill in this space. So you can think of the cytoplasm kind of as like the air in a room. So in this classroom that I'm sitting in, there's all these desks or pencils or computers or whatever is filling up the space in the room. And wherever there's not an object or a person, then you're going to have air filling that space. So inside of the cytoplasm, animal cells will have a cytoskeleton. So this is something unique to animal cells. So the cytoskeleton is basically a skeleton inside of the cell. So if you think about what your skeleton does in your body, your skeleton is your bones. So your bones give your body a specific shape. So you have 2 arms, 2 legs, a head, a trunk, and so it gives your body a shape so you're not like just a blob. So the cytoskeleton in an animal cell is going to give it that spherical shape that animal cells are known to have, which not every animal cell has that. Anyway, so the cytoskeleton is made of, it kind of looks almost like a spider web. So it's a web of protein fibers, and so it holds a cell together, it keeps its shape, and prevents it from collapsing or enfolding in on itself. So some things that make up the cytoskeleton is these 3. The microfilaments, microtubules, and intermediate filaments. So microfilaments are rod-like structures, and I'm going to show you a picture in just a second. And they have different lengths, and they're made up of the protein actin, and they give the cell shape and support, and sometimes they even help the cell move. They also are shock absorbers for certain things. So like pseudopods and amoebas. Amoebas are unicellular, so those microfilaments are going to act as shock absorbers for an amoeba because, you know, they're unicellular, so that means that they're subject to like their environment things can hurt them, whereas your cells are going to be protected by other cells, if that makes sense. Microtubules are going to be more like cylinders, and so they're very straight. They are larger, and they are made of the protein tubulin. So anyway, microtubules also can make up centrioles, which we'll talk about, cilia and flagella. So if you recall talking about cilia and flagella earlier. So these are used when your chromosomes separate out during cell division, when one cell becomes two cells. Another protein, it's called dynin, is a motor protein that allows the bending of that cilia and flagella. So as you can see here, the proteins are important. Then you have intermediate filaments, and they're intermediate because they're between the size of microfilaments and microtubules. So again, they're for structure, because that's kind of the whole goal of the cytoskeleton. And it helps anchor the organelles so that they're not just all over the place. It kind of holds them in place. And it's made of the protein keratin. So animal cells maintain their shape by having which one of these? A cytoskeleton, cell walls, cytoplasm, or chromosomes? So hopefully you chose a cytoskeleton. Now, plant cells maintain their shape by a cell wall, but this question asked for animal cells. All cells are going to have cytoplasm and chromosomes. Which of the following is not a function of the cytoskeleton? So we mentioned cell movement, selective permeability, containing spindle fibers for cell division, or anchoring organelles. So which one of these did we not mention? I'm sorry. The question is not. So which one of these did we not mention? So it's going to be selective permeability. All right. So here's a picture of a cytoskeleton. You really rarely see pictures of a cytoskeleton. So if you take a look at this picture here, the other one's just a cytoplasm, and I think y'all probably are well versed in that. So if you look at this picture here, you can see the cell membrane, and then this is showing a mitochondrion. Okay, let me change colors. All right, so this is showing a mitochondrion. And so here's a microtubule. Notice how it looks kind of tubular. And then you have microfilaments and intermediate filaments. So you have the microfilaments and the intermediate filaments down here. So if you look, it kind of is like a web, and it's holding the ribosomes in place here. It's holding the mitochondria in place, and it's also connecting it to the cell membrane. So this is the cytoskeleton. So these things are going to be like a web, and it's going to help hold those organelles in place. So which of the following structures would you expect to find when examining a bacteria cell? So remember, bacteria cells are prokaryotic. So would you have a cellulose cell wall, chromosomes within a nucleus, a phospholipid cell membrane, or cytoplasmic organelles? So if you recall, bacteria cells are prokaryotic, and prokaryotic cells have no nucleus and no organelles. And I told you all cells have something. Was it a cell wall or a membrane? Hopefully you picked membrane. So all cells have a cell membrane. So that brings us to the nucleus. So the nucleus, all right, is that better? So the nucleus is the brain of the cell, basically. It's the control center, so it acts kind of like your brain does. So it directs cell activities. It stores the DNA. So DNA can come into two different forms. It's not both of these. It's one or the other. And at different times, it's going to be one form, and at other times, it'll be the other form. So the two forms are chromatin and chromosomes. So chromatin you can think of as like spaghetti. It is like unraveled. It's still a double helix, but it's an unraveled. It doesn't make a specific shape. And it also includes some proteins, some histone proteins. Now, one important thing to remember is that you can never see chromatin. When DNA is in the form of chromatin, you cannot see it. Now, the other form is chromosomes. You've probably heard of chromosomes before. Chromosomes are X's, basically. So the double helix of DNA coils into, so think of like a slinky. So the slinky is going to condense and coil into an X shape. When it does that, that shortens the slinky, and it also makes it wider or thickens it. When this happens is when the cell's getting ready to divide. So when one cell is preparing to become two cells. So the only time you can see DNA is when it is in chromosome form. So this is when it's preparing to divide. If you've ever heard of mitosis or meiosis, that would be when it's chromosomes. I just spelled that wrong. So the correct spelling is actually here. Anyway, so two forms of DNA, chromatin and chromosomes. So chromatin, you cannot see it. It's kind of uncoiled, looks kind of like spaghetti. And chromosomes, of course, looks like X's.